botany that had been foreseen by J.C. Bose more than 100 years ago, though they were consistently rejected during his lifetime. Acharya J.C. Bose retired from the Presidency College in 1915, but was appointed Professor Emeritus and was permitted to continue his research work. He gradually shifted his laboratory to his own house, which was situated next door to the Science College in Calcutta. 2 years later, that is in 1917, he succeeded in establishing a research center for himself just north of his house on the Acharya Prafulla Chandra Road, which was then known as Upper Circular Road in North Calcutta. The Bose Institute was founded on November 30th, 1917. He nurtured this institution as its director till his death in 1937. The research center, called the Bose Institute, is set up in buildings designed in the Mauryan style. Like J.C. Bose, the Bose Institute's areas of interest are wide and varied. Though the Bose Institute started with 3 original departments: botany, chemistry, and physics. Today, the Institute is engaged in research in the fields of plant sciences, structural biology, molecular biology, biomedical sciences, biotechnology, quantum mechanics, astrophysics, and condensed matter physics. A Center for Advanced Research on Astroparticle Physics and Space Sciences has been set up at the Darjeeling campus of the Institute. Recent augmentation include the establishment of a national facility on astroparticle physics and space sciences and a center of excellence in bioinformatics. And some 3 hours drive from the main campus of the Bose Institute at Kolkata, this is the Rural Biotechnology Training Center of Bose Institute at a village called Falta. And if you happen to sail for Andaman Islands from the port of Kolkata, you can catch a glimpse of this stately bungalow on the banks of the Ganges at village Falta, where Acharya J.C. Bose used to stay sometimes and reflect. There is also a J.C. Bose Biotechnology Innovation Center at Madhyam Gram and Shamnagar near Kolkata. The Bose Institute is equally recognized today as an important center for training PhD students and postdoctoral fellows. All of what is happening at the Bose Institute today would be quite unimaginable had not Bose dreamt big and worked tirelessly towards realizing them.
जो है जैसा क्यों है वैसा, जो है होता क्यों है होता। हे जो है जैसा क्यों है वैसा, जो है होता क्यों है होता। चमत्कार नहीं सच बतलाए, सिद्ध करके हम दिखलाए। जो है जैसा क्यों है वैसा, जो है होता क्यों है होता। हे जो है जैसा क्यों है वैसा, जो है होता क्यों है होता।
Many years ago, green revolution was ushered in and fertilizers played a pivotal role in it. Chemical fertilizers like urea played a very important role in this revolution. These fertilizers helped in increasing the productivity and took agriculture to new heights.
Whether it is trees or other plants, humans, or any other forms of living beings, everyone needs food and nutrition to survive. Trees and other plants derive most of their nutritional needs from nature, utilizing air, water, and minerals from the soil and the sun's light and heat. But often, special nutritional supplements are needed for fulfilling the special needs of some plant varieties. Fertilizers are such chemicals that helps plant fulfill their special needs. Urea is such a chemical supplement that is often needed to increase the fertility of the soil. It is a colorless, odorless, white, solid, granular chemical compound. In the world of carbon chemistry, urea is also known as carbamide. Being rich in nitrogen, it is an essential soil nutrient. It dissolves readily in water and is absorbed by the soil, making it easy for plants to absorb it and use it for their nutritional needs. That is why it is used widely in agriculture as a nitrogen-adding chemical compound. Urea is used the world over as a soil nutrient for agricultural farmlands. Urea is used also as a raw material in the production of plastics, urea formaldehyde, resins, glues, hydrogen, etc. Urea-based feeds are also given to cattle to supplement their metabolic nitrogen needs.
Not only this, urea is also used to make a medicine called urea stebimine, which is very effective in the treatment of disease called Kala Azar. In fact, urea is also used to make some other medicines also. Not only that, urea is also used in our vehicles as a pollution controller. Who could have discovered this multi-utility substance and how?
German chemist Friedrich Wöhler was the first scientist in the world to synthesize an organic compound from an inorganic compound. The discovery was largely accidental, and he was not attempting to do this, since he too, like other scientists of his age, believed that it is not possible to synthesize organic compounds from inorganic ones outside the body of an animal. On 22nd February 1828, Wöhler was working on some compounds called cyanates in his laboratory. He accidentally obtained urea by treating silver isocyanate with ammonium chloride. This is how an inorganic compound was transformed into an organic one. He then wrote to the Swedish chemist Jöns Jacob Berzelius stating that he had artificially synthesized urea without the use of animal or human urine. Till then, the world of chemistry had been following the theory of Berzelius that organic chemical compounds such as urea could not be manufactured outside the animal body because to do that needs what Berzelius called the vital force of living beings, including humans. It was Wohler who proved for the first time that organic compounds could be synthesized outside a living being. And this is how he proved the earlier tenets wrong and thus laid the foundation of organic chemistry. Following this discovery, urea became a key chemical in developing the chemical industry. This added a huge momentum to the chemical industry. Acharya Prafulla Chandra Ray gave new directions to the chemical industry in India by founding the Bengal Chemicals and Pharmaceuticals in 1892. The company still exists and produces vitamins, cosmetics, asthma drugs and many other important chemicals.
Hey, जो है जैसा क्यों है वैसा जो है होता क्यों है होता Hey, जो है जैसा क्यों है वैसा जो है होता क्यों है होता
Red, yellow, green, blue. All these colors attract us a lot, don't they? But do you know, chemicals are behind all these colors. Take a look.
The myriad colorful objects adorning the shops in our marketplaces attract us easily. Whether these are bangles, clothes or toys, we are surely drawn to them by the beautiful colors and desire to own them. However, the principal factor that draws us to these articles is the colors used in them. Many years ago, natural dyes were used as coloring agents for various things. These used to be made out of flowers and plant leaves and other parts of plants. But gradually, the demand for dyes and colors increased and the supply of natural dyes could not match the demand. That is when the need for creating artificial dyes was felt. And soon, William Henry Perkin unleashed a major new wave within the then ongoing Industrial Revolution by synthesizing the first artificial dye in England. In 1856, Perkin was working on synthesizing quinine from coal tar when he mixed aniline and sodium dichromate. It suddenly produced a rich purple color. Made from aniline, it was commercially named morphine, and Perkin got a patent for this in 1857 when he was just 18 years old. Later on, Perkin also produced two other major colors named Britannia Violet and Perkins Green. By 1862, more than a dozen new artificial dyes had been created, and Perkins' discovery of the first synthetic dyes became a major boost to the textile industry.
Different type of clothes were made available as the textile industry kept growing. Now there was a need for different dyes for different clothes. But why is it so?
Compared to natural dyes, synthetic dyes are much more stable and fast, which means they do not get discolored by washing in water, alkalis and acids. These dyes are also able to withstand higher levels of heat. The atoms of dyes bind to each other due to additive bonds, covalent or valency bonds, hydrogen bonds, ionic bonds and van der Waals force. Dyes can be classified into three categories: substantive dyes, vat dyes and mordant dyes. Substantive dyes are usually used to color cotton, paper, leather, wool, silk and nylon. Vat dyes have been used since ancient times by humans, and in practical usage it is insoluble. These are used primarily to color cotton fabric. Mordant dyes are a kind of dye that need a binder to be used as a fast coloring agent for cotton cloth. Mordants are primarily inorganic oxides or salts which are absorbed by the cloth. It is important to note that many mordants, particularly those in the heavy metal category, can be hazardous to health, and extreme care must be taken in using them. Almost 30% of all dyes used on wool are mordant dyes. Indigo, methyl orange and aniline yellow are synthetic dyes. Acid-based dyes are mostly used to color wool, nylon and silk. Alkaline dyes are used to color paper. Food colors are a special category of dyes which are used primarily to add color to food items. These food colors are industrially produced but use specific methods so as to avoid harmful effects because they directly impact human health.
Hey, जो है जैसा क्यों है वैसा जो है होता क्यों है होता. Hey, जो है जैसा क्यों है वैसा जो है होता क्यों है होता.
Sometime back, such tomatoes were available that would burst even if they were pressed slightly in our bags. But nowadays, such tomatoes are available in the market that would remain unaffected even if they were pressed in our bags or if they fall on the floor. What could be the reason behind this?
In today's world, biotechnology plays a very major role, be it in the field of medicine, food industry or agriculture. It involves the science of studying the active principles and components, as well as the chemical properties of biotechnological entities. Biotechnology is being used to enhance the nutritional quality and pest resistance of many crops. Besides this, major experiments are being conducted to select certain genes of particular species and transferring the genes to other species to produce precisely the quality of crops that we desire. These are being termed as genetically modified crops or GM crops. Some years ago, scientists at the New Delhi-based Jawaharlal Nehru University implanted the genes of amaranth or amdana, a highly nutritious food grain, into potatoes and boosted the protein content of potato to a very high degree. Such kind of research producing highly nutritional food through biotechnology is very useful for poor and developing countries to combat the menace of malnutrition. Scientists have also produced such a variety of cotton that has eliminated the need for the spraying of chemical pesticides on the cotton plants. What the scientists did was to introduce the gene of the otherwise highly poisonous soil bacteria called Bacillus thuringiensis into cotton plants, creating a variety called Bt cotton that was able to resist the attack of a dangerous pest called cotton bollworm. Similarly, the scientists have already developed strains of corn that is pest resistant. New genetically modified soybean varieties have also been developed that can withstand severe pest infestation. Biotechnology has also been successful in producing what is termed as terminator seeds, which produce high volumes of the crop and are pest resistant. However, the problem with such terminator seeds is that they can be used only once to grow that particular crop. The seeds produced cannot be used to grow new plants. There has been strong opposition to genetically modified crop technology and the use of terminator seeds, especially due to the adverse impact such crops may have on the environment. But there is great hope that science will soon be able to get out of this impasse. Scientists are using biotechnology to hasten the creation of synthetic organic compounds. And it is entirely possible that someday in the future, scientists will be able to create artificial blood. If that happens, then the thousands of people injured in accidents or wars may not have to die for lack of blood. It might become possible to treat a part of the body that has become unusable due to disease and replace it with an artificial body part. And maybe someday scientists will create microorganisms that will eat away plastics and clean the environment. As of now, these seem like hopes for the future, but it is truly possible with the giant steps that science is taking to make these possible.
Some cases seem unsolvable, throwing up more questions than answers, leading us down blind alleys only to reach a dead end. But all these mysteries can be unlocked, all thanks to a single wonder molecule, nature's detective DNA. But how does this double helix contain the secret code to all these case files? That's a question of science. Detectives have always relied on biology to fight crime. In 1860, William Herschel, a British administrator in India, first used human fingerprints to stop frauds in pension funds. In keeping permanent records of pensioners, he realized that each fingerprint was unique to the individual and remained unchanged through his or her lifetime. And so fingerprints became trusted investigative tools. In the 1900s, another forensic tool was introduced when Dr. Karl Landsteiner showed that different individuals could possess different blood types. This radical discovery prompted detectives to use blood samples to identify suspects of crimes. But none of these tools were foolproof. Fingerprints could be wiped out or prevented by a simple pair of gloves, and two unrelated individuals could have the same blood type, leading to inconclusive evidence. The question then arose, was there a biological material that would leave forensics detectives with nearly zero doubt? For the answer, they had to go deep into the living body, into the nearly 100 trillion living cells that make up a human being. Inside the cell is the nucleus, containing 46 chromosomes, 23 from our fathers and 23 from our mothers. These thread-like chromosomes are carriers of DNA. And it is here that modern-day detectives can find solutions to their mysteries. Deoxyribonucleic acid, the most powerful molecule in the biological world. Found inside every living cell, it carries the blueprint for every life form. Be it a plant, an animal, or us humans, DNA is the common denominator. The complex molecule of DNA is made of just 4 building blocks or nucleotides. They're named adenine, cytosine, thymine, and guanine. Like partners in crime, these nucleotides exist in pairs. A and T are complementary partners, as are G and C. Together, they form the rungs of the ladder that make up the double helix of DNA. There are over 3 billion base pairs in the human genome itself. That's about half the human population of the entire planet. So what makes this double helix the blueprint of all life? Just like letters in the alphabet combine to form all the words of a given language, these nucleotides or base pairs arrange themselves into genes and non-gene sequences. Genes are fragments of DNA that are responsible for a particular biological function or trait. For example, they determine characteristics like how tall we are or how thick our hair will be. Us humans possess nearly 30 to 40,000 of them, but this only makes up 0.01% of our DNA. The remaining 98.8% is non-coding DNA, or sequences that do not directly correspond to any biological function or trait. For a long time, it was believed that each of us possesses the same arrangement of genes. The idea that it could be different in different people was not common knowledge.
It was believed that all of us have the same genes. There is no difference between different individuals.
But all that was set to change, and it would inadvertently revolutionize forensic science. How? The key is polymorphism. Polymorphism is a common phenomenon in the natural world. It describes biological variations that are common within the same species. For example, all humans have blood, but this blood shows polymorphism. That is, it exists in different blood types. No single type can be designated as the standard. each one, whether A, B, or O, is an acceptable variation. It turns out that DNA also exhibits such polymorphism, and just like blood types, its variations are inherited from one generation to the next. In the early 80s, this startling discovery had geneticists asking, was there a way to observe these variations? Yes, as was discovered by Dr. Lalji Singh, an Indian scientist at the University of Edinburgh in United Kingdom. While studying the sex chromosomes of snakes, he stumbled upon an intriguing sequence of non-coding DNA, or DNA that doesn't directly result in a biological trait. This sequence of base pairs seemed to repeat itself again and again along a specified stretch of DNA. Thanks to this property, it was called simple tandem repeats, or STR. For example, this segment of non-coding DNA contains an STR made of nucleotides GC, repeated 15 times. Another STR shows the nucleotides GCT, repeated 11 times. The fascinating fact is that just like blood types differ from human to human, such STRs also show polymorphic variation. This was an exciting discovery, but its full implications would only be understood in a lab not too far from Dr. Singh's. The year was 1984. Sir Alec Jeffreys had a eureka moment at the University of Leicester in England. He had observed STR variations in DNA samples collected from different members of the same family. These STRs showed variations in different family members. They also showed some similarity. But what did all this mean? It meant that two individuals could be told apart just by studying their DNA samples. Sir Jeffreys proposed that DNA could be used like a fingerprint to conclusively identify an individual as distinct from all other individuals on the planet. Not only that, DNA polymorphism was hereditary, so one could determine parenthood by studying DNA fragments. Sir Jeffreys' discovery revolutionized the world of forensics, and it shone a light on DNA's powerful potential to be a detective. Today we know that humans only differ from each other by 0.1%, and it's this minute difference that provides the clinching evidence.
0.1% may look like a very small number, but in terms of the actual number, it is more than 10 lakh differences.
But perhaps the biggest advantage of DNA over traditional fingerprints or blood samples is its inherent resilience.
It's such a powerful tool, DNA. Is very important because unlike protein, it's very stable. You can boil it, treat with alkali, both the strand of DNA will come apart. The moment you put in neutral buffer, snaps back again, it gives you double-stranded DNA.
1987, back in India, the Center for Cellular and Molecular Biology had caught wind of Sir Jeffreys' revolutionary new findings. Founder-director Professor P.M. Bhargava saw a direct link to Dr. Lalji Singh's discoveries with the snake called banded krait.
He asked me, why don't I develop DNA fingerprinting probe? I was reluctant, saying that the discovery of DNA fingerprinting has already been done by Alec Jeffreys. What is there to develop? He said his probe is patented, is not available. It's going to have a tremendous application.
What exactly was this probe? Dr. Singh had identified STRs, or simple tandem repeats, in the DNA of the banded krait. This STR, GATA, repeated itself 66 times along 545 base pairs. It also showed variations from one individual to another. Most significantly, these variations were observed in humans as well. This made it ideal to be developed into an investigative probe to identify individuals. Once this repeating fragment was isolated, it was coated with a radioactive substance called P32. When it was exposed to other DNA samples, the radioactive GATA segments attached themselves to their complementary partners. If there were any variations between different samples, they would show up on an X-ray film as different lengths of radioactive bands. The probe was called BKM after the banded crate. And in 1988, it made India only the third country in the world to have its very own DNA fingerprinting probe. It was first admitted in the Madras High Court to solve a paternity dispute, but it wasn't immediately accepted. It would take another paternity case in Tellicherry, Kerala, for the first DNA fingerprinting evidence to be upheld in an Indian court. Since then, DNA fingerprinting has come a long way. This is CDFD, the Center for DNA Fingerprinting and Diagnostics. It's where the country's most difficult cases are cracked, all using the detective properties of DNA. It's used to identify suspects in criminal cases. It resolves paternity disputes. It can identify disaster victims whose bodies are beyond recognition. It has even found great use in preventing wildlife crime and food adulteration. It's also used to diagnose prenatal genetic disorders. And where detectives once took 2 months to conduct DNA fingerprinting, today it can be achieved in a matter of days.
DNA technology has reached a level of maturity where everywhere in the world it is referred to as the gold standard of forensic medicine. We reach levels of probability of a match between an individual and a sample that are far more than the number of individuals on the planet, by more than a factor of 1,000,000.
So how do DNA detectives investigate their cases? They need neither criminal records nor eyewitness reports. All they need is a sample to extract DNA from. This can be extracted from just about any biological source. It can even be obtained if the individual is no longer present to provide it. Blood, saliva, semen, skin, hair, teeth, and bones are all powerful sources of DNA, and even small amounts can reveal a lot. But each sample needs to be handled differently in order to extract DNA. They are put through different rigorous procedures to extract the truth-telling molecule.
The way DNA is isolated either from blood or from this is you break open the cells so that the nucleus comes out, and by using different chemical reagents, and then you isolate the DNA. The decalcified bone powder is now mixed with forensic buffer and phenol chloroform for isolating the DNA. After mixing these components, then they are centrifuged to separate both the aqueous and organic phases. After centrifugation, we get two phases. The aqueous phase contains the DNA, and the organic phase contains all the cellular components.
Once the DNA is separated from other cellular debris, it can be studied more closely. In the lab, simple tandem repeats, or STRs, are obtained by adding restriction enzymes to the DNA sample. Just like a scissor, these restriction enzymes cut the strand into STR sequences. These STRs are then amplified or increased in number by millions in a procedure called polymerase chain reaction, or PCR.
A real breakthrough in forensic science came with the discovery of polymerase chain reaction. You know, the biggest problem of forensic science was to get enough DNA from remains, and intact DNA. Those deficiencies were overcome by the technique of polymerase chain reaction, for which Nobel Prize was awarded.
Once PCR amplifies the amount of DNA, it's time to study its STR fragments more closely. After all, they are the real DNA fingerprints of the individual. This is done through a process called electrophoresis. First, the sample is dyed into a distinctive color. Then it is added to a gel called agarose. When electric current is passed through this gel, the DNA begins to split into fragments. Depending on molecular weight, these negatively charged fragments move towards the positive poles. Now the plate is put under ultraviolet light, where the dyed fragments of separated DNA will show up as bands. This sheet of DNA bands is what makes us distinctive, even in a crowd of millions. All that needs to be done now is compare the number and frequency of bands against other samples in order to determine a connection. In 1992, this technique was used to solve the sensational assassination of Rajiv Gandhi by linking blood sample DNA found on site with that of the suspects Thanu and Sivarasan. More recently, in 2012, a decades-long legal dispute was scientifically resolved when DNA fingerprinting proved paternity between politician N.D. Tiwari and son Rohit Shekhar. Today, DNA fingerprinting technology is at its most advanced. However, there are still challenges, especially when the source of DNA is either too little or too damaged. Just such a challenge was presented to CDFD by the Gujarat High Court. In the aftermath of the 2002 riots, mass graves of unidentified bodies had been discovered. Samples of the skeletal remains were sent to Hyderabad for DNA analysis and positive identification.
That was important for two reasons. One, it allowed the investigating authorities to establish that this was indeed a mass grave related to the 2002 riots. The second, it was important from a technical point of view because it challenged us in terms of how we could apply DNA technology to decomposed and degraded remains.
Then, in 2010, CDFD faced its biggest challenge yet. A devastating air crash along a steep slope of Mangalore's airport had claimed over 150 victims. Many bodies were beyond recognition, making positive identification extremely difficult. Once again, DNA stepped in as detective. Currently, CDFD's research teams are working on developing technologies where DNA is either in short supply or badly degraded.
To get a successful profile from those degraded samples is a challenging exercise. So now we are looking at different technologies like single nucleotide polymorphisms to help in human identification.
Single nucleotide polymorphisms, or SNPs, are the most common genetic variations found between two individuals. Here, there may be a difference of just one nucleotide in a specific stretch of DNA. This single nucleotide variation can help link an individual to a sample, even if the available DNA is very less in amount. But DNA technologies don't just solve headline-making cases. They can also bring comfort to families grappling with crippling diseases. For many families afflicted by genetic disorders like physical and mental developmental issues, heart disease, or cancers, etc., DNA fingerprinting can help cope with the situation and plan for the future.
All these genetic disorders can occur throughout right from birth of the child to an adult, and whenever they have a problem, either they consult us directly or they are referred to us by a physician. So then we examine them and we decide what test needs to be done, and then these genetic tests help us to identify the reason.
We can counsel the family, and if they wish to have one more child, obviously the parents are worried, will that child also have this error? Will that child also have this disease? How will we be able to cope with it? So before a child is born, when the mother is pregnant, we are able to do certain tests to ensure or to check whether the unborn fetus, the unborn child, has that error or not. And if that error is there, then the parents can be counseled. Would they like to have the child or undergo an abortion? So a therapeutic abortion, as it is.
A single molecule of DNA, a myriad of complex cases. As a life-giving molecule for all things in nature, its purpose is much bigger than just human dilemmas. And so scientists at CDFD turn to DNA to safeguard one of India's signature products, basmati rice. This fragrant, flavoursome and elegant grain is a celebrity in the rice kingdom. Only grown in this part of the world and highly sought after by many, basmati exports are often victim of a terrible crime, adulteration. Luckily, DNA detectives are there on the scene.
We identified some of the DNA markers which were specific to basmati rice. Using these specific markers, we can actually detect non-basmati rice in the basmati rice samples. So adulteration level detection can be as precise as 1 to 3% level. When we presented this protocol, and also we published these protocols, and we also have a patent on this protocol.
Most recently, DNA fingerprinting has moved on from protecting flora to protecting fauna. Conservation groups around the world and in India are increasingly turning to DNA to fight crimes like poaching and illegal trafficking of endangered species. The wonder molecule of DNA is not just an accurate detective, but also a versatile one.
Now the question is, what is going to be the future technology? Now latest development which is taking place from genomics to phenomics. You have a DNA scene of crime. Can you sequence the DNA and make the picture of the criminal who has committed the crime? So you don't have to go anywhere. The DNA can tell you the whole story.
Nearly 30 years after first being used in forensics, DNA technology in India has come a long way. It has cast its lens beyond sensational court cases to encompass a higher goal.
20 years ago, when the technology was first introduced, there would be questions in court discussing, particularly from the defense lawyers, discussing, is this technique valid enough, is it accurate enough? Those questions no longer apply. Future of the technology, I would say, is really in expanding its use within India. With a very conservative estimate, we are doing less than 1% of cases that needs to be done, right? And I think our goal will really be to expand.
With these challenges to meet, DNA detectives forge ahead with renewed vigor. In the coming years, they hope to tackle a far wider variety of complex mysteries. Thanks to the double helix, many more case files will then be conclusively closed. If you'd like to share your feedback on today's program, please send your suggestions and comments to Vigyan Prasar, C24, Qutub Institutional Area, New Delhi 110016, or you can mail us at info@vigyanprasar.gov.in.
Long, long ago, when humans were still hunting and gathering, totally dependent on nature for all their needs, exploring the world around them, learning from trial and error, learning from plants and animals. Hidden from their sight, under the ground, lay a knotted hand. Brown as earth, no beauty to attract, no color to lure. Yet, we dug it out. This is the story of the wrinkled truth, ginger. Ginger, the humble spice, taken for granted by most of us. In reality, exceptional. A spice with an ancient history, fascinating stories and personalities ranging from the forefathers of medicine to monarchs and queens. Ginger, native to India, has earned for itself the name Maha Aushadhi, the great medicament. Taken fresh as adrak or used in its dried powdered form, sont, ginger is an incredible spice.
Traditionally, ginger is known as Vishwa Bheshaj or universal medicine. Why it is termed as universal medicine? Probably because it contains so many molecules and every molecule has a different role in your body.
Shringaveram, like the horn of a deer. The name ginger has roots in Sanskrit. Zingiber officinale, scientifically, for us simply the pungent spice ginger. An ancient spice native to India and China. The use of ginger goes back thousands of years. Thou shalt be made to drink a cup, the admixture of which shall be ginger, says the Holy Quran. In Ayurveda, ginger is an entire medicine chest, recommended as part of every meal. Chinese philosopher and teacher Confucius ate a bit of ginger before each meal. He said, "Do not take away the ginger. It aids health and digestion." Ginger has earned for itself the title, the crop of truth, a hardy plant cultivated extensively in India, from sea level to an altitude of 1500 meters above. It thrives under heavy rainfall and even with irrigation. The plant produces erect, tall and dark green leafy shoots. Its flowers pink and attractive. A major cash crop, ginger grows underground.
Ginger as a botanist is very innovative structure, because if you are talking about pungency, the pungency which is present in the rhizome will keep all the kind of predators, pests and diseases away from the ginger plant so that the next generation can come up.
Ginger's protective mechanism, its pungency, has become a potent spice, dug out after 7 to 9 months of growth underground. Families, no matter how different, share resemblance. The leaves of these 3 cousins reveal the lineage. Ginger, turmeric and cardamom, 3 spices from the large Zingiberaceae family. Ginger enjoys a very special place in the Indian spice box. The box that very often doubles up as a medicine chest for wellness. Taken with honey or lemon or plain salt, this is shared wisdom. how did humans discover spices? How did different sets of people living in different disconnected lands reach similar conclusions? The wondrous evolution of humans. To gain an insight, we reach a nowhere land. A land of people suspended in time. In the heart of central India, surrounded by the tall Satpura range, lies a deep valley. Patal Kot, believed to lead to the mythical Patal Lok, hell, is remote and inaccessible. Cut off from main civilization till a few decades back, Patal Kot is home to Gonds and Bharyas, tribes who live amidst nature, depending on nature for health and wellness, with an intimate knowledge of plants.
यहाँ तो 5 पीढ़ी से ज़्यादा हो चुका होगा सर, मैं 5 पीढ़ी तक तो मैं जान रहा हूँ। हमारे जो आजा पराजा जो भी सियाने लोग बुजुर्ग थे वे बताकर वे चले गए, ऐसा है सर। मैंने मेरे बच्चे को मैं बता दूंगा, वो देख के वो भी करने लग जाएगा, ऐसा है।
Knowledge gained from years of experimentation, passed down generations. Plants have always been a part of human medicine. These medicine men, known as bhumkas, depend heavily on local plants. Their knowledge and skill acknowledged by seasoned experts.
जंगल से लाकर अपने खेतों में भी लगा लिया है सर, तो वहीं से फिर उखाड़ के वो खांसी सर्दी जुखाम कफ सांस होता है तो वो खून जाम हो जाते उनको साफ कर देता है।
A wild pungent variety grows in this green valley. Ginger, smashed fresh and steeped in black tea, is a remedy known all over Asia. Sukhan Singh's son is unwell with fever and congestion. He too will be drinking this brew for the next 3 to 4 days. This folk medicine, the only remedy available to him here. They hope it'll bring relief. With no doctors visiting the valley, these people have no easy access to medical facilities even today.
तबीयत खराब है, बुखार चढ़ा। हाँ, जड़ी बूटी वाले तो हैं, मैंने डॉक्टर के पास जा रहा है।
It's a difficult, secluded life. Medicinal plants like ginger help bring relief, but not always. The song underlines the importance of the visit to the outside world, to the bazaar. Once in a while, the villagers have to climb the steep hills to shop for essentials and mingle with civilization. For the nearly 2,000 natives of Patal Kot, this is the only link with the outside world. When they emerge to buy essentials, they bring with them their skills and knowledge, a knowledge of medicinal plants and their use. This is how knowledge of spices would have traveled in yesteryears. Their flavor, aroma, and goodness, making them precious and desirable.
ये पथरी के लिए अलग अलग पेट दर्द के लिए है, पेट मरोड़ के अचानक पेट दर्द होती थी, इनको फल्ली पीस के पिला दो।
In this weekly market too, the lure of spices is evident. Spices that have enticed humans with their unique properties, literally carving and shaping the world and diet. 200 years before Christ, there was a trading city in India named Shunti. 130 years after Christ, Galen, the Greek physician, was using ginger as a purificant of body. In yesteryears, a pound of ginger could buy a whole sheep. Such was its value and demand. During the 5th century, ginger plants were grown in pots and carried on long sea voyages between China and Southeast Asia. It helped sailors counter seasickness and the deadly scurvy. The Portuguese slave owners of West Africa fed ginger to their slaves, hoping it would ensure a stronger population. For them, it meant a larger profit. The faith in ginger's healing capacity has been universal and handed down generations. It's a single spice used in various forms and combinations.
खांसी जुकाम कुछ भी हो तो अदरक की चाय पी लो, गले का बहुत फ़ायदा होता है इसमें। सब्जियों में डाल देते हैं, पेट में हवा वायु वगैरह नहीं होगी, सूप में डाल देते हैं, सब चीज़ों में हम लोग अदरक डाल देते हैं और दवाइयों में तो बहुत ही फ़ायदा करती है।
Ginger, a warming spice, is universal medicine in Asia. In traditional knowledge, where all disease is a result of imbalance in the body, ginger is a chosen healing spice.
From the beginning of the Indian civilization, medicine is practiced as a part of home. Our culture is, who is the best doctor at home? Grandma.
Pregnancy, accompanied by the first 3 months of discomfort with nausea and vomiting. Since generations, ginger with lemon is the remedy women have turned to.
अदरक और नींबू थोड़ा खाती थी मैं प्रेगनेंसी के 3 महीने में, क्योंकि नौशिया के लिए बहुत अच्छा रहता, मतलब जो गैसीनेस है वो निकल जाता तो थोड़ा रिलैक्स फील होता था.
Fresh ginger and lime in the early stages of pregnancy, and now after the baby's arrival, dried ginger, sont, has stepped in. A sweet halwa laced with the pungent dried ginger, something most Indians across regions would identify with.
और माँ ने भी मतलब मुझे